You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech Health Podcast. And I have Younginger. He's a postdoc researcher at University of Washington in the Department of Plant Pathology. So, Brett, thanks for coming. How you doing?
I'm doing well, thanks. Thanks for having me.
Yeah, so tell me, I didn't even know there really was such a thing as plant pathology, but what got you interested in plants in the first place, and then what led you to your current focus?
Always been a plant lover. That's something that I've connected with inherently throughout my entire life. Strangely enough, had a sunflower garden when I was 6 or 7 years old that I planted and tended, and spent a large portion of my childhood wanting to be a farmer in some form or another. And then throughout high school and college, just have always had an interest in gardening and plants and the natural world, and all sorts of plants, not just garden plants, but trees and mosses, and as well as fungi. So yeah, it's always been something that's there. And I got an undergrad degree in biology, not knowing what I wanted to do, and just kept pursuing what I enjoyed, and that led me to where I am now.
Okay. So what's your current research about?
I'm currently in a group that studies plant microbial symbiosis. Specifically, we study leguminous plants, so think peas, beans, in the plant family Fabaceae. So these plants form symbiotic relationships with bacteria in the soil that are able to convert nitrogen in the atmosphere, that is the most abundant gas in our atmosphere, but it's in an unusable form for plants. So they can convert that atmospheric nitrogen into a plant usable form, plant fertilizer, essentially, through a process called nitrogen fixation. And they do this in the nodules of on the roots of the plants themselves. So we study all sorts of things related to the ecology and evolution and the theory of these nitrogen fixation symbioses. But my background is more in plant fungal symbiosis. I worked for my dissertation, for my PhD, worked on fern host systems, and then studied fungal endophytes. This is a class of fungi that live entirely within plant tissues, and when you are observing them, they don't cause symptoms of disease. And they're in all land plants studied to date, so important to keep that in mind next time you're having a salad, that all plants host fungi inside of their tissues, not just bacteria.
Wow.
But yeah, so moving more into the bacterial end of things since I've been in this postdoctoral position, but it's been a lot of fun studying some new systems and doing some really exciting research.
So how does nitrogen fixation work? What are some of the steps in the process?
Yeah, sure. So it's atmospheric nitrogen, as I've said, and this has to permeate through the soil. And then also through the root tissue of the plant where the bacteria are housed in these nodules. And then the bacteria are able to cleave the dinitrogen bond, which is a triple bond, really hard to break, an energy-intensive process. And then they reduce that nitrogen into ammonia, which the plant can then use. So they need to carry out this process in hypoxic or very low oxygen conditions in order for it to occur. And again, very energy intensive. And it's a mutualism because it does benefit the plant. Nitrogen, if anybody's gardened or grown tomatoes, it's a good time of year to discuss growing tomatoes, right? In the garden, your plants are not going to grow if they don't have enough nitrogen. That is the most limiting macronutrient in the soil. So this process is critical to both terrestrial natural ecosystems and to agricultural systems as well. Think the crop rotations that farmers implement when they plant soybeans in between corn. That's in an effort not only to produce soy, which is widely used, but also to increase the amount of nitrogen in the soil.
So the soil, when nitrogen permeates soil, does it act at all like a liquid? The nitrogen, is it just in the interstitial spaces in the soil particles, or does the nitrogen somehow, I don't know, join with the soil and become in a different form instead of just gaseous nitrogen?
Yeah, I should answer this question with a caveat that I'm not a physicist or a soil chemist in any regard, but my understanding is that it does permeate as dinitrogen, as N2. It's not entering in a form that is altered because it's very stable as dinitrogen because of that triple bond.
Right. Yeah, I read about like the Haber-Bosch process to take atmospheric nitrogen and make ammonia from it, and that's like very high temperatures and pressures, but yet these bacteria do it without those things.
Right.
How well understood is the mechanism by which they do it, and can that be replicated in the lab?
The Haber-Bosch process, and then the process of nitrogen deposition through lightning are the other alternative methods by which nitrogen ammonia can be created from nitrogen gas. I am not that well versed in the fine scale mechanistic understanding of the process, but I know it is well studied, and it happens through an enzyme called nitrogenase, and that enzyme itself is what is sensitive to oxygen. So it can oxygen can inhibit the enzyme from cleaving that trinitrogen bond, and then further inhibit the reduction into ammonia. I'll leave that to the biochemists. That's certainly my area of expertise. Critical process to the green that we see on our planet. Were it not for this process, our planet would not be as green as it is. Suffice to say.
Where is the focus? Go ahead.
Leguminous plants exist. They're near cosmopolitan. They have a near cosmopolitan distribution. It's a very large and successful plant family. So, think acacia trees. I mentioned things like beans and peas. Those are more cultivated plants, but certainly they have wild counterparts as well. Things like clover and medics and all sorts of plants, and they're all over the world and really can facilitate the existence of other plants that need that nitrogen to grow. So, these plants will oftentimes be some of the earliest to colonize, and they'll build up nitrogen. In disturbed habitats, will be the first to colonize. They'll build up nitrogen in the soil, and then other plants can come in after them and make a living.
Oh, so they don't use all the ammonia that's produced by the bacteria, and a lot of it just stays in the surrounding soil, or how do the other plants get nitrogen from their activity?
So eventually, when those plants die or senesce, those nodules can slough off into the soil, and then they will deposit that nitrogen in the soil.
Oh, okay. So there's stores of large amounts of nitrogen that's available, and then they go into the soil, and then they become part of the matrix. Okay.
Yeah.
So what's your specific area of study then in the nitrogen fixation process?
Currently working on a few projects. One of which we're studying the role of partner choice. This is a process called partner choice that both plants and bacteria will undergo in order to associate with different strains of nitrogen fixing bacteria. And when I say strain, suffice it to say like a different, not species, but for now, we can just say it's a different individual bacterium, right? A different strain of bacteria. So, certain bacteria are better or worse at undergoing nitrogen fixation. And we're interested in how plants are, if they are, and how they are discerning what is a better nitrogen fixer and what is discerning better from worse in the soil, right? Because if these plants could discriminate between better and worse strains of bacteria in the soil, it would provide a fitness benefit to them, which would in turn, they would leave more offspring, and it seems like that would be adaptive. So that's something we're interested in. We're currently conducting some greenhouse studies, examining how readily this process occurs, and I guess.
Well, how do you know that it does occur? Like when you looked at, let's say, I don't know, a particular kind of, pea plant, do you see that there's a wide range of different strains of bacteria that do the nitrogen fixing for them? And do you see that within a small area that's planted? Like certain plants prefer certain kinds, or is it uniform? Like what have you seen?
Right. So these strains oftentimes are naturally occurring. I mean, farmers do inoculate with strains of bacteria, but they're free living and naturally occurring. And plants do get colonized by multiple strains simultaneously. And some plants are better at being colonized by just a few strains. And then also the bacteria, I should say, are playing a role in this as well. Some are really effective at colonizing plants, and then some are not as effective. And then, there's not always perfect alignment with the benefit that they're giving to the plant. So the really beneficial strains aren't always the best at colonizing the plants as well. So there is quite a bit of variability, and how that variability plays out and its importance in both agricultural and natural systems is really important to understand. So farmers can inoculate with nitrogen-fixing bacteria, but if there's other bacteria, native bacteria in the soil that more readily colonize the plants, they can, and they might not necessarily be better at performing nitrogen fixation, they can colonize the roots instead of what the farmers are applying to the field, and they call this the competition problem. So even though these farmers want to utilize and leverage nitrogen fixation, oftentimes it's not as successful as they'd hoped because there's native bacteria in the soil that aren't as good at the process, but will still colonize.
Are you able to inoculate a plant and give it the bacteria you want? And I mean, let's say a seed, are you able to plant something along with the seed to make it more likely that it'll have the nitrogen fixing bacteria you want, even if it's planted in soil that has bacteria that are not as good that normally would take over?
Yeah, so if you're planting it in soil that is not sterile, it's certainly more difficult to control that. So in our research group, we do inoculations with single strains of bacteria and recover just those strains of bacteria because we're doing it under sterile conditions. So that we can create readily. It's harder to control in non-sterile conditions, or if you're doing multiple strains simultaneously. And that is one component of this greenhouse project that we're currently working on, is inoculating with multiple strains that we've fluorescently labeled with proteins that will fluoresce under certain wavelengths. And then we can image those and see what strains are present, how many nodules they formed, how big those nodules are, and then in turn see how well the plants are doing with these respective strains.
Are there situations where there's an intermediary between the soil and the nitrogen in it, and then, maybe there's one bacteria that starts the process of fixation or does it, and then it passes, some of those metabolites onto another bacteria that actually is in the nodules of the plant? Do you see any chains of cooperation that go on, or is usually just one bacteria that's the intermediary between the soil and the nitrogen and the plant?
Yeah, I mean, it's I don't know the number off the top of my head, but there's so many bacteria in just 8 grams of soil. It's a really competitive community that exists around these plant roots, and these plants are exuding carbon compounds that the bacteria need to survive into the soil. So it's basically drawing bacteria towards them. So there's lots of competition that's happening for both these plant sugars, this plant carbon, and also for the process of colonization. And then even though these bacteria, it may be a predominant strain of nitrogen-fixing bacteria that are in a nodule, there will also be other types of bacteria that co-colonize. And those are certainly so the plant, in return for getting this nitrogen, it's not a one-sided relationship. The plant will return carbon and sugars even more so to these nodules where the nitrogen-fixing bacteria are housed. But other bacteria that don't fix nitrogen are able to exploit this and coexist in these nodules with the nitrogen-fixing bacteria. So that's a whole other layer upon that.
Right. Have you tried an experiment where you have your sterile soil, and on the left, you put a plant in there, and on the left side you put strain A, and on the right side you put strain B, and see which one the roots preferentially grow towards. I don't know if there would be time or if the strains would quickly go through the soil so that there's no time for a decision. But have you ever been able to create a situation like that where you're seeing if the roots somehow are sensing the type of bacteria that they want and they're growing towards them to meet up with them?
It actually happens in the opposite. The bacteria are modal in the soil and responding to signals, those sugar and carbon signals. They're really flavonoid compounds or carbon-based compounds the plants are putting out there. So the bacteria are responding to what the plants are exuding into the soil or putting out into the soil, more so than the plant growing towards the bacteria.
Well, I heard that roots seek moisture and they can sense it through the soil matrix. And maybe they are doing some kind of reciprocal stuff.
Yeah, potentially. I'm not sure if that's known. So maybe that's a new future research direction that we should check out. But yeah, plant roots are certainly responding to sources of nitrogen in the soil too. So I mean they're carrying out foraging as well.
Is it possible to put a plant and have split up the root bundle so that half of the roots go into one type of soil that's isolated from the other half of the roots and see what that does? Like you preferentially water one side and you put certain bacteria in one side. And, have you done that kind of stuff?
I have not. Split root experiments is what they're called.
Okay.
But they are notoriously difficult, but they are conducted. Regarding root morphology in response to nitrogen-fixing bacteria inoculations, I am not familiar with research that's investigated that, but that does not mean it doesn't exist. But yeah,
okay.
I think at the end of this podcast, I'm going to check it out and see if it does exist.
Excellent.
I suppose I should give you acknowledgments if a publication results from that.
Oh, I appreciate it. Within individual nodules, are you seeing the composition of bacteria is very different, or is it pretty uniform throughout a whole bunch of nodules?
Regarding the co-colonization of different types of bacteria that are in there, is that what you mean by uniform? Or
whether there's if you look at two nodules on two different roots, they're all in the same soil. Will they have the same set of bacteria in them, or is it different types of co-colonization that goes on in different nodules? For some reason, maybe certain nodules are closer to the heart of the root bundle and others are further out, and therefore they preferentially give out different compounds and attract different types of bacteria. Have you seen that?
Yeah, sure. I mean, they are communities, and they do vary throughout the soil based on who is in the soil, who is near, and also, as you've said, where it's located physically on the plant. So yeah, I mean, these nodules do vary quite a bit in size, morphology, shape. They all tend to have roughly the same shape, but you'll look on an individual plant and see that some nodules are really large, some have many lobes on them, some are really small, some tend to be up near the top of the root, some tend to be down near the root tips, which would be deeper down in the soil. So there's a lot of variability there. And then certainly when you look within the nodules, so we do DNA sequencing of nodules to see what bacteria are present there, you do see some variability. And that is really context dependent on what was in the soil ahead of time and what was most competitive. So that's also an active and interesting area of study, not just for us, but for several research groups, talking about some of the factors that might affect who ends up together in a nodule, and depending on who's in that nodule, how effective nitrogen fixation is.
Do you see that do multiple nodules act in concert? I know in the plant they do, because the plant's all one organism, but the bacteria that hang out in each nodule, they're not a biofilm, but do you think there's communication between the nodules and a network effect that's going on where their bacteria are somehow communicating with each other across nodules and communicating what's going on?
That is another good question that I don't have the best answer for. I know that, I guess in science, we shouldn't be surprised if something like that happens. I know that roots are responsible for transporting minerals in the soil and water obtained from the soil up to the shoots. Meanwhile, shoots are sending sugars through photosynthesis back down to the roots. I'm not sure how much internodule interaction is occurring, though, but presumably if there is multiple nodules on the same root, and the flow is traveling along that route. That same flow is passing those nodules. So potentially there could be some communication, something like quorum sensing that may occur. I don't know if you're familiar with quorum sensing in biofilm formation, but I'm not sure how readily that happens and how that affects the process of nitrogen fixation. And it's important to keep in mind that these bacteria are performing nitrogen fixation because they proliferate abundantly in these nodules. And then when those nodules senesce, they're able to seed their offspring, their progeny, into the soil. So it gives them a big fitness advantage, even though it's really energy intensive to form these nodules and carry out nitrogen fixation. So they're able to propagate themselves much more readily because they're getting all those plant sugars, which has led to their success. But so I think of that in a competitive sense, right? Because they're all trying to, there's strong selection for them to propagate their own DNA. But I don't know, I'm not aware of sort of like a collective signaling that's happening in different nodules, because I tend to think of those as kind of distinct communities of bacteria that all have their own interests in mind. But again, suppose I wouldn't be surprised if there was signaling that it was happening on an internodule basis that was affecting both the process of nitrogen fixation and then the bacterial fitness.
Is there a chicken and egg scenario? Like, what comes first, the nodule or the bacteria? Can you tell which one is signaling the other?
What was that? Say that last part.
So, I mean, can you tell that does the plant form the nodule expecting bacteria to take up residence, or do the bacteria inform the plant, hey, make a nodule?
Yeah, it's a molecular dialogue. It's a multi-factor authentication, to borrow from my boss's saying. So the process of forming a nodule requires plant signaling and then a bacterial response to that signal. The bacteria need to be close to the root, and then through multiple molecular steps, the root hair itself starts to deform and actually kind of wrap around the bacterium, and then it kind of encases the bacteria within the plant tissue, and then that plant tissue will kind of continue to divide and grow and create this housable space that is of low oxygen, that is suitable for the bacteria to carry out the process. Also, there's a lot of overlap between plant pathogen signaling. So plants need to be able to discern pathogens in the soil. Now, bacterial pathogens are not as widespread and abundant for plants as fungal pathogens tend to be, but still plants are antagonized by bacterial pathogens and fungal pathogens. So a lot of the same signaling that happens in pathogen defense for plants has been co-opted for this process of nitrogen fixation. So there's an abundance of overlap in these signals. So it's presumed that it evolved from initially from pathogen defense or pathogen signaling from plants.
And are the bacteria in a nodule temporary guests, or are they permanent? And do they refurbish the nodule once they've been there for a while and change it?
No. So when they get colonized by this strain, this bacterium that serves as sort of the seed for, because lots of propagation or cell division happens in these nodules after it gets colonized by a strain of bacteria. And again, during this process, though, other bacteria can colonize. But it's not like there's, to the best of my knowledge, there's not a lot of turnover between strains once this nodule is formed. It's more that it gets colonized by a strain or two strains simultaneously. That also happens, or multiple strains. But then once that process happens, it's more that the bacteria are reproducing within the nodule to both increase nitrogen fixation and also ensure their success once that nodule kind of floats off into the soil, and then they can repeat the process again.
So what would be a really happy result of your research? What are you trying to figure out?
Ultimately, I'm excited by basic science, but applied science doesn't happen without the legwork of basic science. Oftentimes people are just curious about the natural world, and they're trying to understand the patterns that we see when we look outside. But and then people can utilize that basic information and make applications in agriculture and technology and so forth. So at a fundamental level, I'm really excited by understanding these processes that tend to impact to a large degree the patterns we see in nature. But also really closely related to that is if we understand these patterns, how plants are discerning between different strains of bacteria in the soil, this can definitely help us leverage using nitrogen-fixing bacteria in agricultural conditions, which is of very high need because we have soil erosion and water pollution happening from our current fertilization practices. So there's a very strong interest in leveraging nitrogen fixation, naturally occurring nitrogen fixation in both legumes and leguminous plants, ones that already are able to carry that out, but also transferring that to other plants that don't naturally carry out nitrogen fixation. So there's a big push from the Department of Agriculture, USDA, to understand how we are potentially able to transform non-nitrogen fixing plants into nitrogen fixing plants. So like cereal crops, for example, wheat I know is one that they're interested in seeing if we can transform them and make them nitrogen fixing plants. So, I mean, understanding how plants are able to discriminate between different strains of bacteria in the soil would go a long way towards engineering plants that could potentially carry out nitrogen fixation. Although they,
what would be the benefit of a legume, for instance, doing nitrogen fixation so that other types of plants can grow in the same area it grows? Is it doing it to help the other plants? Is it just like a coincidence that it does?
No, it's got its own best interests in mind. It's important to remember that. But yeah, it's so when nodules do naturally senesce off of the roots, that becomes available for other plants. But for the time being, when that plant is existing in that space, it is trying to hold its own ground and just obtain enough nitrogen for its own growth and reproductive success. Yes, it facilitates the existence of other plants, but that's from a natural selection standpoint, that's not why the plant would do that. The plant's performing nitrogen fixation for its own success, not for other competitors. It's a cold world out there in the natural world. So kind of like
what's the
every
best,
individual's in it for themselves.
Yeah, I got you. What's the best nitrogen fixer you know, and combined with the best nitrogen fixing bacteria? Like, are there any super fixers out there?
Yeah, I mean, well, we have strains that tend to be really good at fixing and other strains that tend to not be so good. And that's an interesting question. How does that play out in natural communities? Why does that even occur? Why aren't all strains really good nitrogen fixers? And we do have some understanding of that through both theoretical and empirical work as to why some bacteria are really good and some are bad. And also from the plant perspective, some plants are really good at discriminating amongst different strains of bacteria. And then plants also are able to cut off the flow of oxygen and potentially nutrients to bacteria after they colonize the roots, and if they're not doing a good job of nitrogen fixation. So we do know that as well. So yeah, but we do see, I don't know about individual plant species, leguminous plants, which happen to be really good at forming nodules versus different strains. But the plants that we work with, we work largely with clovers, trifolium, but naturally occurring ones, not forage ones, wild clovers we work with, and also with medicago, which is a classic nitrogen fixing plant that is widely utilized for research. But yeah, we see broad variability in different genotypes, different genes, different plants that are able to form these nodules, and then different strains as well. Yeah, that variability is very intriguing. And again, that would go a long way towards pairing up leguminous plants with the best bacteria. So if we could figure out some of this, the partner choice that I was speaking to earlier, if we can better understand that, it's more likely we'll be able to pair up certain genotypes of plants with the best strains of bacteria.
Yeah. Okay. Well, very good. What's the best way for people that are interested to learn more? Should they go to like the lab website or university website, or should they contact you directly? What do you recommend?
Yeah, they should check out our lab website. It's symbiomics, like symbiosis, only omics, symbiomics.com. We're the Friesen Lab here at Washington State, so definitely check us out. And there's a lot of interest around this process, obviously in agricultural settings and just natural settings. People are really intrigued by this process because of how important it is. So there's lots of information out there, and it's not all really dense science information, science heavy writing. But yeah, and I think the Gates Foundation just funded a startup that is trying to leverage nitrogen fixation in non-traditional plants as well. So it's very trending field of research because there's a growing human population that we need to feed. And in order to do that, we're going to need more nitrogen. And the nitrogen is going to have to come from more sustainable forms. And that form is through this process.
Well, very good. Well, Brett, thanks for coming on the podcast. I appreciate it.
Yeah, thanks for having me. It's been a pleasure.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.